Now let's take a look at enzymes. So here's your overview. Enzymes are defined as catalysts that speed up the rate of a chemical reaction without altering the result. So they catalyze reactions that would occur naturally, but it would take forever to complete the reaction. So they speed up a reaction without altering the result, and they are not altered in the process themselves, so they can then just be recycled and convert another substrate molecule into a product molecule. So important thing to know is they're catalysts, and the reactants that they interact with, they're called substrates. There are isozymes, so think of them as families of enzymes that do similar things. For example, amylase. There's a salivary amylase, and then there's a pancreatic amylase. So both of them are amylases. They're just in different places, and they have slightly different condition preferences, but they are isozymes, so they work, they're pretty much cousins of each other. So they catalyze the same reaction, but under different conditions. So one in your mouth, for example, the other one in the small intestine. Enzymes, they may be activated, inactivated, or modulated. So let's take a look at it. Some of the vitamins, they become coenzymes, so they are necessary for the enzyme to be active. Without the coenzyme, the enzyme would not function properly. When we say coenzyme, then we mean an organic substance, for example, a vitamin. So all the B vitamins, for example, they're important for your energy metabolism. Those are coenzymes, or something that works together with your enzyme, like NADH or NADPH, those kind of things. Then we also have cofactors. So listed here are cofactors. They're inorganic molecules such as an iron molecule or a zinc, or the ones that you typically find on your supplement bottles as minerals. So that would be cofactors. Let me add this right here, cofactors. And the example here, iron or zinc. So those are inorganic, they're smaller, inorganic. Then we have chemical modulators such as pretty much the conditions, such as pH or the temperature preference that a certain enzyme has. So here is a sort of a sidetrack here. These are important enzymes for diagnostic purposes, to diagnose certain diseases. So here an example given. Let me skip down here to the middle. This one is very important. Creatine kinase. The creatine kinase level gives an indication, or is indicative of whether somebody had a heart attack. So myocardial infarction is a heart attack. Also muscle disease, because muscle contains a lot of creatine kinase, and if muscle tissue is damaged, then it gets released and you can pick up that enzyme in the blood. Here's another one that's you may have heard about, glutamate dehydrogenase, liver disease. But I think the one that you may have should have heard about is creatine kinase, the CK test. This is for myocardial infarction. So let's just leave it at that. This is just a sidetrack here for you to take a look at for some diagnostically important enzymes. Okay, so let's take a look at conditions under which enzymes want to operate. So the enzymes, they speed up a chemical reaction. They interact with the substrate and then gently lower the amount of activation energy that's necessary for the reaction to occur. They all are pretty picky. Most enzymes that work in the human body have a pH optimum of 7.4, right around where your blood pH is. So here will be 7.4, and you can tell, so it's about 7.4 right here. And you can see that at pH 5, the rate of activity here, enzyme activity, would be near 0. And if you're close to 9, the rate is 0. So this enzyme here, or these enzymes, the ones that are working in the human body, they typically have a pH preference of 7.4. That's the blood pH. So, and here is in general what enzymes do. So here we have our reactants. Those are our substrate molecules, the starting compounds for the reaction. This here, the dotted line, is the amount of activation energy that's normally required for this reaction to occur and produce products C and D. So without enzyme, we have to overcome this much activation energy. The chances for this to happen is really low, and that's why you get a really low rate of this reaction occurring naturally. Now, let me take this off here again. With the enzyme, we're going to follow this nice little hill right here, and it's so much easier. So the enzyme will temporarily lower the amount of activation energy that's necessary. So now we only need this much energy, and that's much easier to achieve. And so now the rate of the reaction is going to speed up because it's so much easier for these molecules to go up that little hill and then react, and we're going to have our product C and D. So this is pretty important diagram here that you should definitely understand. So here, of course, on the y-axis we have the energy bar basically, and down here is our timeline. So I hardly can see that. Okay, so make sure that you understand that graph. And this table right here, we have just some sort of a summary of different types of reactions. The ones that you will, well, for this lecture, you're gonna see a lot of redox reactions or oxidation reduction reactions, and then all the digestion reactions are hydrolysis reactions. So hydrolysis, dehydration, synthesis kind of reactions. So making proteins, that would be a dehydration synthesis, or making starches, dehydration synthesis. So these two types of reactions are by far the most important. So redox reaction, oxidation reduction reactions, and then hydrolysis, dehydration synthesis reactions. These we already covered in a little chemistry lecture. So since in this lecture here, we're going to talk about aerobic respiration, let me say a little bit more about oxidation versus reduction. Oxidation reduction involves the transfer of hydrogen, electrons, that is. So most people intuitively would say an oxidation is, oh, I'm adding oxygen, and that is true a lot of times, but you can also oxidize something by removing electrons or hydrogen. So in general, oxidation reaction means that we're adding or subtracting electrons, and they sort of form these hydrogens. So a reduction means that we're gaining electrons, we're adding hydrogens, that is, or we are losing oxygen. That's the alternative way of defining reduction. So again, oxidation, intuitively most people would say, oh yeah, I'm adding oxygen. That's maybe true, but a lot of times it's actually the removal of hydrogen what happens, and reduction is the opposite. We're adding hydrogen, we're losing oxygen. Okay, and here are some examples: dehydrogenase, reductase, these are all, or oxidase or oxidoreductase. These are all different enzymes or enzyme groups that will catalyze redox reactions. And then here, hydrolysis, dehydration, synthesis. So we have all of these hydrolases that will hydrolyze something, hydrolysis reactions. Here's some more examples given. Peptidases, lipases, all of the digestive enzymes are in this category. And I feel like the other ones, you will really not have any contact with this. In the context of this course, we really don't need them. Okay, so let's move on to defining metabolism. Metabolism is the sum total of all the chemical reactions that are taking place in an organism. That's a big word, and really metabolism is sort of this all-inclusive, all the reactions that are taking place. We can break down metabolism into those reactions that release energy versus those that require energy input. So catabolic metabolism, or catabolic reactions, or here abbreviated as catabolism, that's the part of the metabolism that generates products that have lower energy amounts than the original substrate molecules. So we're going downhill on the energy scale. Good example for that would be breaking down glucose to CO2 and water and gaining some ATP. That's catabolism for sure. Or also breaking down proteins into amino acids, or carbohydrates into simple sugars. anabolism, that is the part of metabolism where the products have more energy than the initial substrate molecule. So you're building higher energy molecules. Good example for that is protein synthesis. You're adding amino acids and you're making proteins out of it. And the protein molecule has more energy than the individual amino acids that you put in. Okay, another term that you definitely need to know is kilocalorie. Those of you who speak Spanish, you know what calor is. Calor is heat. So a calorie is a measure of heat or energy that is being released. So when you take a look at food labels, you always just see, you know, how many calories something has, and those are actually kilocalories. The way a kilocalorie is defined, it's if you were to convert the energy content of food, a piece of food, a piece of bread or whatever it is, a peanut or marshmallow, if you were to set that on fire and will burn it down completely to CO2 and water, and using the heat that's released from burning down that food to warm up some water, then here's your definition for kilocalorie. It's the amount of energy that it takes to heat up 1 liter of water by 1 degree Celsius. Or if you want to go with the small calories, it's the amount of heat that's necessary to heat up 1 milliliter of water by 1 degree Celsius. Okay, so here's your kilocalories. So now you know. When you read the food labels, think of the heat that's being released if you were to burn that food by setting it on fire, actually. Now, our body's actually pretty good at burning down just metabolizing the food and also generate some heat and some energy out of it. Anyway, if you are setting a marshmallow on fire and then you're going in one-step reaction, so there's nothing in between. The human body, we dare not to do that because that would be pretty bad. So we tend to gently lower or release the energy that's, let's say, in a carbohydrate. And for that, we need whole pathways. So we need pathways with typically many, many intermediates to gently release the energy step by step, not in one big combustion reaction or in some big explosion, because that would be not too healthy. There is a diagram in your book that I'm not too fond of, but it kind of does explain sort of what's going on. So here you have a roadmap, and then over here you see a metabolic pathway sort of showing what the steps of a certain metabolic pathway. And you can see how glucose gets converted to glucose 6-phosphate and then fructose 6-phosphate, blah blah blah. So anyway, so these metabolic pathways you can see step by step, and it kind of does resemble a roadmap, if you will. Now let's talk about how cells regulate their metabolic pathways. Of course, you can control or regulate a pathway by controlling the enzyme concentration. So that's a good way to do that. You can produce modulators that change the reaction rate, such as inhibitors that are usually temporary or reversible. You can use different enzymes to catalyze reversible reactions. So that's one good way to regulate. And then you can compartmentalize enzymes within organelles so that the enzyme reactions are only taking place in certain places. And you can maintain the optimum ratio of ATP on AD to ADP. So all of that would do that. And here's a very popular way of how cells regulate their metabolic activity. So here you have a metabolic pathway of A to Z. So we have lots and lots of steps in between. And basically, so we need, for every step, we need a different enzyme. Enzyme 1 will get us from A to B. Enzyme 2 will get us from B to C, and so forth. Now, once you have enough of Z, then you just somehow want to stop the reaction. So a lot of times, Z becomes an inhibitor to enzyme 1. So now, once we have enough Z, then Z by itself becomes the inhibitor to an early enzyme in a metabolic pathway. And that process is known as feedback inhibition. So let's move on to take a look at some reversible metabolic reactions that are controlled by enzymes. Example A shows you how we have a reversible reaction. And I told you earlier that's very important for our pH homeostasis. The conversion of CO2 plus water to carbonic acid is completely reversible and depends on what your body needs. Both steps are catalyzed by carbonic anhydrase, and so here we have one enzyme for both directions. In this case here, we have two enzymes for this reversible reactions from glucose to glucose 6-phosphate. We're going with hexokinase. And from glucose 6-phosphate back to glucose plus phosphate, we're going to go with glucose 6-phosphatase. So that's just another example. And here we have on panel C, we have this irreversible reaction that there's no back arrow. So this would be, has only one direction. And there's only this one enzyme listed the same as over here. So, okay, so let's take a look at ATP since that's so important. And that's what we want. That's what the cells want all the time before we then head into the glycolysis, the Krebs cycle, and the electron transport chain. So here, let's take a look at the ATP transfer of energy. And ATP is considered the energy currency of the cell. It has this very high energy phosphate bond, and specifically it's the bond between the second and the third phosphate. So ATP is adenosine triphosphate. And when we release the energy, we split off that third phosphate and make it adenosine diphosphate, ADP. So that bond between the second and third phosphate has just a lot of energy, and it can be easily transferred by taking that third phosphate off, and then you can transfer that energy that's released to power other reactions. Okay, so now, and the nice thing about ATP and ADP is it works like a rechargeable battery. So you can easily convert the ADP back to ATP by putting some energy in, and you recycle your ATP. So I think we're ready now to take a look at aerobic metabolism. This is how your cells gain energy, the energy that they want. They want lots and lots of ATP, and they do this in a 3-step procedure that's known as aerobic metabolism or aerobic respiration. The first step is glycolysis. The second step is called the citric acid cycle or Krebs cycle. And the third step is called the electron transport chain. And yeah, I'm just going to write this down, Krebs cycle. That's alternative nomenclature. And keep in mind, the reaction that we're following here is we're going to take a glucose molecule as our model molecule. So we're going to take C6H12O6 plus we need to breathe in oxygen. We don't need that oxygen. And we are going to convert in many, many steps that glucose molecule to 6H2O plus 6CO2, and of course these have very low energy. Water and CO2 has very low energy, so the energy that was in the glucose, then we get an energy output of about 36 ATP. That's pretty good, actually. So we're going to take a look at how this works. I'm just going to start a new recording to not make it too long. At the end, we're also going to take a look at anaerobic metabolism. That's basically just involving glycolysis, because without oxygen, you have to stop at glycolysis. For the Krebs cycle and the electron transport chain to occur, you must have oxygen. It's very important. So anaerobic metabolism, it gets you a little bit of ATP out of glycolysis, but it's really not enough to run our metabolism in the long run. So our cells rely on aerobic respiration for making enough ATP for all the cellular processes. All right, so to be continued in the next recording.